Hi, my name is Dustin Wolkis, and I'll be presenting on the potential advantages and pitfalls of using living collections as a living laboratory, and then share a couple of real-life examples. So if we look at botanical institutions worldwide, we'll find almost 1.5 million collection records, representing almost 600,000 taxa and a total of 3,600 institutions. And these can be used as an excellent resource in seed conservation research. It takes pressure off of collecting from wild species, and oftentimes they're well documented with well curated data. And it's more easy to carry heavy equipment to the garden and to outfit plants with instrumentation. They can be used in citizen science, and you can turn them into an outreach and education tool. And it's a great example of staff from many different departments all working together. In general, it's more easy to collect from the garden, and you have a semi-controlled environment. Permits may be easier to obtain or unnecessary. Plus, you have a coffee bar and a toilet just around the corner. So now I'll share a couple of examples. The first are these two subspecies of Hibiscus waimei. Subspecies Hanneri is critically endangered, and subspecies waimei is near threatened, and both are single island endemics, endemic to the island of Kauai. And it's thought that these species have physical dormancy, which is characterized by a water impermeable seed coat, and so no imbibition can occur. And what do I mean by imbibition? No, not that kind of imbibition, but this kind where water enters the seed. It turns out that that's pretty easy to measure, because as water enters the seed and the seed imbibes, they become heavier. And so we think that water is entering through the chalazal gap or chalazal slot. And so we went out to the McBride Garden on the south shore of Kauai, and also to Limahuli Garden on the north shore, and measured imbibition in seeds that were scarified, boiled, soaked, and an untreated control. And it turns out that the control did imbibe, and so by definition, they are not physically dormant. And maybe that's a surprise, but maybe not, because these other two species of hibiscus, including Tiliaceus, which is indigenous to Hawaii, are also not dormant. We also looked at germination, and it turns out that the control performed as well or better than any treatment. So these taxa do not require any kind of dormancy breaking pre-treatment for uniform and successful germination. The next example I'll share is from Poliscius racemosa, or formerly Monroidendron. And it's thought that this species can either be morphologically dormant or morphophysiologically dormant. In morphologically dormancy, embryos can either be undifferentiated or underdeveloped, and therefore need time to grow inside the seed prior to germination. And you can measure this by looking at the ratio between the embryo and the seed. And so if morphologically dormant, the embryos will grow inside the seed over time. And so we went out to McBride Garden and harvested seeds and herbarium specimens. And we found that indeed the embryos do grow inside the seed prior to germination. So it has morphological dormancy. But we also looked at physiological dormancy and found that an alternating temperature regime of 25-15 degrees C was enough to overcome physical dormancy. Also, 500 parts per million addition of gibberellic acid could speed up germination. So Poliscius racemosa has morphophysiological dormancy. However, there are a couple of potential pitfalls in using this kind of data. Specimens can sometimes be misidentified and lacking provenance data. You can be missing different types of interactions. You could get unintended hybrids that never would happen in nature. And you could get site-specific physiological responses just as a byproduct of being grown in a different environment, growing in different soils, or the addition of supplemental water, as shown here, and nutrients. You can get genetic erosion, which is the loss of genetic diversity. You could also get artificial selection, especially by harvesting seeds grown from garden plants. And so as long as you can mitigate these types of pitfalls, then using living collections as a living laboratory is an excellent resource. Mahalo.